Nitrosamine risk assessment for the pharmaceuticals. Nitrosamine are the impurities which have the carcinogenic and mutagenic potential, and these impurities have a very stringent limit at ppm level. So nitrosamine impurities are very difficult type of impurities to handle in the pharmaceutical formulations, in the pharmaceutical APIs, and in the excipients. I have made many videos on nitrosamine impurities, but still I want to make the videos, and I am continuing to explore the information because every time uh, we need some updates, we need to refresh our knowledge about these topics. Welcome to the video, and let's start with some introduction for the risk assessment. Nitrosamine impurities are considered as cohort of concern. These have potential carcinogenic uh, activity, or you can consider these impurities as potential carcinogens. So these are required to be controlled at a very low level. And regulatory bodies like USFDA, EMA, WHO, ANVISA, and many more are requiring or require the stringent control over nitrosamine impurities. Nitrosamine impurities are required to undergo the risk assessment activity so that you can identify, mitigate, and control the impurities in your drug substances, drug products, and excipients as well. Nitrosamines are two types of impurities, which are small type of nitrosamine impurities, and some are like NDSRIs. So the nitrosamines which have structural similarity with the API are called as NDSRIs, and the small nitrosamines are also there. Risk assessment of the nitrosamine involves risk assessment factors for the APIs, excipients, API synthesis, manufacturing process for the drug product, packaging material, and packaging processes. API synthesis requires reviewing the manufacturing process, and the risk assessment is involved in the secondary and tertiary amine presence, and also the presence of nitrosating agents. So if these secondary or tertiary amines are present, then these will increase the risk. If nitrosating agents are there, it will increase the risk. Excipient are required to be assessed for certain nitrosating agents or amines, and manufacturing procedure is required to be evaluated for conditions like acidic environment, high temperature, and use of the solvents. So because these type of environment promote the nitrosamine formation, then packaging material are also required to be assessed for the risk. Identify leachable impurities like nitrosamine for rubber stoppers or nitrocellulose containing packaging. This is not the only information or only points for risk assessment. Other points are also there. Then evaluate the potential pathways for nitrosamine formation like API process impurities, which may be formed during the synthesis or maybe because of the degradation. Excipient and API interaction may lead to formation of these impurities. Reaction between the nitrite impurities, amine groups in the excipients. Sometimes nitrosamine get formed because of the storage and stability of the pharmaceuticals. Nitrosamine formation due to product decomposition or long-term storage may also happen. Then the another stage is or step in the nitrosamine risk assessment is perform the analytical testing. If you found that the nitrosamine may be present or it is likely to be present, then develop a sensitive and validated method to detect the nitrosamines at low levels. The methods are required to be very sensitive because the nitrosamine impurities are controlled or are required to be controlled at a very low levels like parts per billion range. So LCMS, MS, GCMS are the methods, some of the methods to detect the nitrosamines. Screen for the common nitrosamines like NDMA, NDA, and some others, and confirm the results with the stability studies under accelerated condition. Then the risk categorization. So risk categorization is always like high risk, medium risk, and low risk. High risk means presence of known amine groups or nitrosating agents. If you know that, or if you conclude that there is a presence of known amine groups or nitrosating agents, then it will be a high risk product. Medium risk, possible contamination from excipients or solvents. Low risk, absence of risk factors in formulation and process. Then mitigation strategies. There are various mitigation strategies based on the formulation and based on to the risk assessment. Reformulation involves replacing amine containing excipients or APIs with safer alternatives. Process optimization involves controlling the reaction conditions like pH temperature to minimize nitrosation. Supplier qualification involves ensuring the excipients and APIs for meeting stringent nitrosamine limits, and packaging changes involves use of nitrosamine free materials. Establish the control strategies, set acceptable limits, AI levels specified by regulatory agencies. Monitor nitrosamine levels during routine quality control and include nitrosamine testing in the stability protocols. Always understand that if the product is at moderate risk or high risk for the nitrosamines, it becomes very important to control the nitrosamine limits below the regulatory control limits or threshold limits. Then documentation and submission is required. Prepare the risk assessment report for nitrosamines and involve the risk assessment in total. Consider all the points and keep it ready for the regulatory submission. Now see the practical considerations. Limit of detection, or it is abbreviated as LOD. It ensures methods can detect nitrosamines below regulatory limits. For example, 26.5 nanogram per day for NDMA. Cross contamination risks are there also. Review potential contamination risk in multi-product facilities. If the facility for manufacturing manufactures the products which are prone to risk or which are risky for the nitrosamine, then consider the cross contamination risk for other products also. Regulatory compliance is the another challenge. Follow agency specific guidelines like ICH M7 FDA IMA guidelines for the nitrosamine risk assessment, nitrosamine testing, and nitrosamine uh, limits and compliance. Example of the nitrosamine risk assessment workflow is included here. So initial screening is there for preliminary review of synthetic pathways, excipients, and packaging. Then analytical evaluation will involve test the batches for nitrosamine impurities. If you come to know that there is a risk for nitrosamine impurity formation or nitrosamine impurity presence, then some batches are required to be tested for these impurities. Risk mitigation involves implementing the process for formulation or formulation changes for the findings, and then control strategy is required to be made and stringent limits are required to be set for the nitrosamine impurities. Also consider nitrosamine risk and NDSRI risk as well based on to the API structure, based on to the excipients, and based on to the uh, manufacturing process of API, manufacturing process of the drug product, storage and stability conditions, packaging material and packaging processes. Also, you can connect with the regulatory bodies so that you can take information and help from them to set the limits. Also, you can use the interim limits provided by the regulatory authorities till you get the NDSRI or nitrosamine limit finalization. Then one of the thing is that uh, whether the nitrosamine is coming from API, whether it is due to the excipients or nitrite impurities or nitrosating agents in the excipients, or it is coming because of the process. So all these parameters are required to be evaluated very critically. This is the information for nitrosamine risk assessment in the pharmaceuticals. I have made many videos on nitrosamines. and NDSRIs. You can go through the playlist for complete understanding about these impurities. Thank you for watching the video.